In this model, I'll be exploring cell replication, and I'll be focusing on answering the following questions. What are the processes involved in mitosis and what is its outcome? What are the processes involved in meiosis and what is its outcome? And what is the importance of cell division for the continuity of a species, and how does this differ between the two processes? Replication is the process by which cells replicate their genetic material and divide to form new cells, which is vital for growth, repair, and reproduction. Cell replication can occur through either mitosis or meiosis. Let's begin with the process of mitosis. In mitosis, one original parent cell divides once to produce two identical daughter cells. Mitosis is important in multicellular organisms as it creates new body cells needed for growth, repair, and maintenance. This increases the chance for survival by ensuring the organism grows to reach an age for reproduction. Without mitosis, a zygote would never be able to transform into a complete adult organism. Let's take a deeper look at how this works. Before mitosis, each cell starts off with a full set of chromosomes, making it a diploid cell. The first step of mitosis is DNA replication, also known as interphase. This begins with the replication of DNA in the form of chromatin, which is the parent cell's DNA wrapped around proteins located within the nucleus. Next, while two identical chromatin pieces are still joined at the middle, they quickly shorten and thicken to form a chromosome, two chromatids connected by a centromere. This stage is called prophase. Metaphase is the stage in mitosis where chromosomes line up individually along the midline of the cell. Next, organelles called centrioles that float around in the cell move to opposite ends of the cell. The centrioles then release protein structures called spindle fibers. In the next stage, spindle fibers bond with the centromere of each chromosome, shorten and pull the chromatids apart and towards them on either side of the cell. This is called anaphase. Finally, in telophase, the cytoplasm pinches off and separates. This is called cytokinesis. The cell and nuclear membranes reform and the chromosomes uncoil to create two diploid daughter cells with identical genetic information and the same number of chromosomes as the parent cell. Let's look into meiosis. The process of meiosis creates gametes that carry out sexual reproduction, allowing genetic material to be passed on while maintaining the correct number of chromosomes in organisms. Meiosis can be categorized into two parts, meiosis 1 and meiosis 2. We do this because during meiosis, one original parent cell divides twice to produce four non-identical daughter cells with half the amount of chromosomes as the parent, making the daughter cells haploid. Meiosis also allows for the continuity of a species as organisms can produce offspring with new gene combinations, creating a genetic variation as a result of recombination due to crossing over. This increases the likelihood for survival in the case of sudden environmental change. Let's begin with looking into meiosis 1. DNA replicates in the form of chromatin. The chromatin begins to shorten and thicken to form chromosomes. Next, the chromosomes line up in homologous pairs, one chromosome inherited from the father and one from the mother that code for the same genetic information. These pairs line up in a random order called independent assortment. Crossing over then begins to occur. Homologous chromosomes swap sections to produce new gene combinations, increasing the genetic variation among the daughter cells. As the membrane around the nucleus breaks down, centrioles release spindle fibers that attach to the centromeres, shorten, and homologous chromosomes break and move to either end of the cell. At this stage, the centromeres don't break like mitosis. The cytoplasm pinches off and separates, and the cell splits into two and division occurs. This two begins with chromosomes lined up individually along the midline of the cell. The centrioles on polar ends of the cell release spindle fibers that attach to the centromeres of the chromosomes. The spindle fibers shorten, the chromosomes break, and strands move apart to opposing sides of the cell. The cytoplasm then separates, cell and nuclear membranes reform, and chromosomes uncoil. This marks the end of meiosis. It was just amazing at visualizing scientific processes and concepts, simplifying complex ideas, and building an understanding that can be communicated. However, they can be limited. Let's analyze the success and limits of this model. This model uses simple terms and diagrams to explain and visualize cell division. This can be advantageous. However, this can also be disadvantageous as it oversimplifies the dynamic nature of mitosis and meiosis. Chromosomes typically are more complex than displayed in this model. They are 3D structures folded in highly specific ways, which is not visible. Similarly, during meiosis, it should be evident that one homologous pair differs in size and shape from another homologous pair. However, they have been shown to be the same size and shape. Whilst it accurately describes the concept of independent assortment of homologous pairs, it does not display the random orientation of homologous chromosome pairs during meiosis 1.